All right, welcome everyone to this lightning talk session. We've got 5 presentations. These are all video recordings, but I do see some of our speakers are online, so we'll hold questions at the end, and hope you enjoy these presentations. So the first one is from Jill Jacobson, and I'll press play for that now.
Hi, my name is Jill Jacobson, and today I'm going to share with you the results of the first study from a grant that Dave Hauser and I received from SSHRC. As you might recall, in the reproducibility project psychology, cognitive psychology studies had a higher replication rate than did social psychology studies. And there are a variety of differences between these two areas. One is the design typically used. So cognitive psychology are more likely to use it within subjects design than is social psychology. And within subjects designs in the reproducibility project psychology also had higher replication rates than did social psychology studies. Another difference between the two areas that often doesn't get mentioned is that the amount of information provided prior to the study also can differ between the two areas, with cognitive psychology often being very explicit about what their research hypothesis is, whereas this tweet by Andy Luttrell captures in social psychology, we tend to have cover stories and do feel it's not right to reveal what our hypothesis is before our study begins. So there have been a variety of studies looking at non-naïveté, and these have found mixed results, anything from less likely to find the effect, so decreasing the effect size, to no difference, to actually enhancing the effect or an increase the effect size. Unfortunately, these different studies that have looked at participant non-naïveté have differed in too many different ways for us to be able to determine what might account for these discrepancies. So in our first study, what we wanted to do is take advantage of the archival data provided by the Reproducibility Project of Psychology. And what we did is we coded the information that was provided on OSF from the various studies. And we looked at the consent forms as well as the instructions and materials. And we used a coding system that basically ranged from 0 to 5, where 0 means no information was revealed, to 5, it was revealed in great detail. Now, there are various ways to assess replication that I don't have time to get into in a 5-minute talk. I do have a link here to a YouTube video of a talk I gave for the Durham Rights Club that goes over all of these in more detail. I'm just going to look at the 3 different replication assessments that were used in the reproducibility project psychology. So was the p-value less than 0.05? Did the effect size fall within the replication confidence interval? And was the meta-analytic effect size greater than 0 or different from 0? So when we look at the consent forms, unfortunately, besides asking specifically and being told that they were available, it turns out only 32 of the 100 studies posted their consent forms on OSF. And you can see that the way that you assess replication does have an effect. One of the big effects that it has is in terms of how many studies would be considered to be successful replications. So it ranges from 9 to 12, depending on which one you use. And as you can see, although this data is very weak, and again, we need to be cautious because it's based on an N of 32, there does seem to be some indication that maybe having more information in the consent form actually would lead, is associated with the study being less likely to replicate. Now, when we look to the materials, now 86 of the 100 studies had materials that were available that we could access and that we could translate. And so again, not all 100 studies as we had hoped. And again, we don't see a whole lot of difference here based on the type of replication assessment that was used, because everything is non-significant. But if you just look at the direction of the means, it does seem that maybe it's the opposite of the consent form. Again, cautious because we don't have significant results here, but maybe more information is associated with a higher replication rate, again, just based on the instructions and materials. So there's some indication here, very preliminary, that maybe it makes a difference where the information is. Unfortunately, it's very inconclusive because of the lack of materials that we have and the consistency of what was provided in the materials. So some are just pictures with no instructions. So in our current research going forward, what we're going to do is actually manipulate the amount of pre-study information and also where it's located to see if we can better understand what's happening here. So thank you for your time. And I just wanted to acknowledge the undergraduate and graduate students who helped us with this research.
All right, very interesting work. I will now move to the next talk, and we can take some time for questions at the end. This one is from Kayana Zahiri on evaluating study design, rigor, and preclinical cardiovascular research.
Hi. My name is Kayana Zahiri, and I'm a senior undergraduate BSMB student at Brown University. Today, I'll be presenting some work I did with a team led by Dr. Adrienne Mueller at the Stanford Cardiovascular Institute about evaluating study design rigor in preclinical cardiovascular research. Here we have a translational research paradigm from the NIH that depicts critical stages of the biomedical translational research process that starts from basic sciences research to preclinical to clinical research and implementation, and ultimately impacting public health. Preclinical studies using animal models play an important role in evaluating the safety and efficacy of novel therapies and ultimately optimizing patient care. They are also important for a general understanding of disease. Yet we often find failed translations from bench to bedside, and methodological rigor remains a major priority to maintain a level of consistent reproducibility across preclinical research. One means to enhance reproducibility is to increase the robustness of study design elements, or SDEs, such as randomization, sample size estimation, blinding, and sex as a biological variable. In this study, we aim to evaluate the inclusion of these key SDEs in preclinical cardiovascular research studies. By identifying these trends, we will assess the rigor of scientific practices carried out in preclinical cardiovascular research. The study that we performed was actually a replication study of Ramirez et al. 2017, which looked into methodological rigor in preclinical cardiovascular studies published between 2006 and 2016 in 5 major AHA journals. We aim to reanalyze these trends that we looked into in the most recent decade and in a wider range of journals. So diving into some of our methods, we performed a PubMed query with the search string you can see below for preclinical cardiovascular research articles using animal models published between 2011 and 2021 in 9 high-impact scientific journals listed here. The search returned 309 articles that we systematically screened for 4 main SCEs. 1, the use of both sexes in study subjects. 2, use of randomization. 3, use of blinding. And 4, performing a sample size analysis. Studies that included both human and animal experiments were screened separately for SDEs in animal experiments versus SDEs in human experiments in the study. To ensure accuracy and consistency in screening, each article was separately screened by 2 individuals, and discrepancies in screening were resolved by consensus. So here we have some of our preliminary descriptive results that show 61.7% of studies screened had animal-only experiments, while 38.3% of studies had an animal and a human experiment component to their study. In terms of publication year for the studies, we had a roughly similar proportion of studies from 2011 to 2021. For the journal the studies were published in, around 45% of studies we analyzed came from PNAS, followed by around 20% from the European Heart Journal, around 14% from Journal of American College of Cardiology, and the remaining 20% or so of articles came from other journals, including Nature, Science, Cell, and The Lancet. For the species used in the studies, we can see the breakdowns in the chart on the bottom right-hand corner. Here we saw that around 2/3 of studies used mouse models and 15% used rats. And then we had a wide range of other animals such as pigs, rabbits, and more. Several of the studies also used more than one animal model organism in their studies. In terms of other results, here we show the counts and percentages of STE inclusion with separate analyses for animal-only studies versus studies that have both animal and human experiments. For biological sex of study subjects used, 60% of animal-only studies reported using both male and female subjects, while 51% reported using only one, and for human and animal studies, 34% reported using both male and female, 8% reported using only one, and over half of the studies actually didn't mention the sex of study subjects used. We found that inclusion of randomization and blinding was quite low overall for both animal-only and human and animal studies. There was also a considerable gap in reporting of randomization and blinding animal-only and human and animal studies, with 33% randomization and 29% blinding in animal-only studies, compared to only 7% randomization and 12% blinding in human and animal studies. Lastly, in terms of sample size estimations, inclusion of this STE was consistently low, with 8% of animal-only studies and 2% of human and animal studies performing statistical sample size estimations. We also looked at the change in the proportion of STEs over time across all 309 studies, regardless of whether the study included human subjects. The data has a lot of variability, but if anything, we see that the proportion of STE inclusion in preclinical cardiovascular research between 2011 to 2021 actually decreased for 3 out of 4 STEs, with the exception of inclusion of both sexes. These trends, however, were not statistically significant. In terms of our conclusions, similar to Ramirez et al. 2017, we found that our preliminary results show limited inclusion of key study design elements in preclinical cardiovascular research. Although not statistically significant, the proportion of studies including both sexes increased between 2011 and 2021, while the proportion of studies implementing blinding, randomization, and sample size estimation decreased. More rigor is needed in methodological design of preclinical cardiovascular studies to ensure high-quality results that can be safely translated down the bench-to-bedside paradigm for clinical research. Further research will be conducted to analyze reporting of STEs over time across journals, differences in STE reporting between large and small animals, and more. Here are our references, and we'd like to acknowledge our Stanford Mavericks research team, including Dominique Patel, Isaiah Jimenez, Gabrielle Montenegro, as well as the Stanford Cardiovascular Institute, and thank you to the AIMOS conference for allowing us to share our research.
Hey, thank you very much. Pretty scary to see reductions in blinding and randomization over time. But we'll move on to the next presentation by Janaynne Carvalho de Amaral.
Good afternoon, everyone. My name is Janine, and I'm a postdoctoral research associate at the University of Illinois, Urbana-Champaign. And today I will present the results of my research on patients in peer review. And by the end of this presentation, you will understand how a health and social care academic journal is engaging patients in the peer review process. I have been studying the history of scientific journals and the peer review process since 2018, and when I started my PhD. And I realized that scientific journals typically disseminate content produced, edited, and evaluated by researchers. However, open science initiatives such as open access, citizen science, and open peer review are expanding the role of peer-reviewed journals. And the goal of this study was describe the methodology used by the journal Research Involvement and Engagement, launched in 2015, to engage patients and the public in the peer review process. And they also discuss the role of this new type of reviewer in the peer review process. And to do this, we collected from the journal's website its aim and scope, reviewer guidelines, and peer review policy. We also collect the editorials present in the journal, and we also interviewed the editors-in-chief of research involvement and engagement. So the results showed that the journal reviews guidelines are the main tool for engaging patients in the public in the peer review process. And the journal review guidelines ask reviewers to consider aspects of the manuscript, such as objectives, methods, results, and ethics. And these guidelines ask reviewers to consider whether the writing is accessible to the reader, and also if there are improvements that could make the manuscript more applicable to the community. In terms of the role of the patients as reviewers, the results show us that academics reviewers and patient reviewers have the same role in the peer review process, and their voices have the same weight in editorial decisions. And exactly as academic reviewers do, patients reviewers may evaluate manuscripts based on their experience and background. And also you can ask yourself, who are these patients? And these patients can be scientists and non-scientists. It's important to remember that sometimes academics are also patients. In addition to engage the public in the peer review process, the journal also is adopting open identities. It means that authors and reviewers know the identities of each other, and the journal adopt peer review reports. It means that open reports. It means that reviewers' names are published in the peer review reports. And also, the results show that these patients are more than reviewers in the journal. In fact, they can be editors, peers, and authors of manuscripts. And the results show that the goal of research involvement and engagement is to go beyond to engage patients in the peer review process. In fact, the journal is co-producing the journal with patients, academics, policymakers, key stakeholders, and service users, before engaging patients in the public at all stages of health and social care research. And as a conclusion, I would like to highlight that the strategy of research involvement and engagement to engage patients and the public in scientific journals may have to build a bridge between scientists and society, turning journals into tools for public engagement in science. And in addition, as a consequence, patients and the public may impact the way researchers do, evaluate, communicate, and publish research results. Here are a few references to conduct this that I used to conduct the research, and thank you for your attention.
Great, thank you very much. Now move to our next presentation by Dragan Okanovic.
Hello everyone, my name is Dragan Okanovic. I'm the founder of Unfold Research, and today I'll briefly cover how you can use Unfold Research browser extension to help you navigate the academic landscape and also help your peers by posting content that could be useful to them and speed up their own research. The problem that we're actually solving is of disconnected research. It's actually quite difficult to discover something high relevant, high quality out there, because if we are in a relatively niche research field, then we are heavily reliant on search engines picking up very obscure papers and other pages and kind of making the connections for us and suggesting them as we are typing their queries. On the other side, if we are in a research field that is heavily saturated, we are faced with a lot of noise and we can't be sure what really is there of highest relevance for us, because it relies mostly on the machine heuristics to suggest the content for us instead of direct social input. It's also very hard to extend existing work because when we post new research, we usually post it somewhere else and not directly in the existing work, which means that finding those two connections is extremely difficult. And also because a lot of these algorithms rely on references and simple reference counting, any kind of gray literature and micropublication format and the rich media format is heavily discouraged. They simply don't have the place in the current academic system. So we'll lose any and all of the benefits that they offer, like very brief summaries that we can very easily consume, etc. So we're very lacking there. And the solution that we come up with is the browser extension that acts as a tool for linking academic research together. With this browser extension, basically any web page in any paper can have the ability that you can post comments, that you can vote on all the comments, leave reviews, upload files and data, and also like link other comments, link other research, and make bidirectional connections also to videos, code repositories, and all of that you can subscribe for updates and bookmark. And so this is a very brief overview, but let's also see a demo. And the problem that we will be facing even today is, where do we find the resources from today's conference? And the best sort of answer would be, well, on the conference website. And with Unfold Research, you can do exactly that, regardless of if you're an admin on the conference website, you can actually link extra content on top of it. So in this demo, I basically visit AIMOS conference website, and right there on the page for anybody else to find who is using the Unfold Research extension, I'm posting slides from this presentation so that anybody else can find them. And on that content, they will be able to post their own comments, ask questions, etc. So we spawn really like an entire array of social interactions right there. In this next demo, what we can also achieve is to create an open review system. Basically, because we have the ability to post reviews on any web page in any paper, we can simply start replying to other people's reviews or whatever other content they posted. We can review their reviews. We can post notes on reviews of reviews, etc. So we basically start spawning really tree of different content that is connected amongst each other, and just providing extra context for all of the research and for any and all of the papers that we might be interested in. All of that can also be voted on, and that kind of helps us just distill the content as quickly as possible and know what is of highest relevance. This in the background, invisible to us, basically starts creating the scientific knowledge graph that we were talking about, where all the different pieces of content for each of the different papers start linking amongst themselves and creating a very semantical scientific knowledge graph that we'll be able to soon completely utilize and not just browse. And on what's next, we are working hard on data enrichment and creating an SDK so that other people can also extend Unfold with any additional functionality and features. We're working also on a web search UI so that you can browse the content even without the extension, and also open API so that then other tools can start consuming our data. We also have some secret what's next categories. So if you're interested in that, definitely browse for these slides and author notes on the conference website. With that, you can find the browser extension on unfoldresearch.com website. It's completely free to download and use. And with that, thank you for your time and happy unfolding.
Very cool. Who doesn't love a secret? So we'll definitely be exploring that. And then our final talk is from Kathleen Schmidt.
Hello, my name is Kathleen Schmidt. I am a research scientist at Ashland University and an associate director for the Psychological Science Accelerator, or PSA. Today, I will be presenting an overview of an exciting new project recently launched by the PSA. First, I will summarize the inspiration for and goals of this initiative. Then I will describe opportunities for researchers to join as collaborators. Research in psychological science tends to rely on convenient samples, typically undergraduate students from universities in Europe and North America. This practice has been described as the field's weird problem, where samples from Western, educated, industrialized, rich, and democratic populations are used to make general claims about human psychology. Research relying on these samples may have low generalizability across cultures and contexts. That means that the results may only hold for a specific subset of the population of humanity. Despite growing recognition of this problem, little progress has been made in expanding sampling or narrowing claims. The Psychological Science Accelerator, or PSA, was established in part to improve the generalizability of research in psychology. The PSA is a distributed network of researchers in the behavioral sciences from across the globe. Our more than 1,300 collaborators represent 84 countries from all 6 populated continents. Research implemented through the PSA network typically provides much more regional and cultural variability than typical psychological science. By pooling our collective resources, the PSA is able to create massive data sets from dozens of countries. Ashland University, in collaboration with the PSA, has received a $975,000 grant from the John Templeton Foundation to provide generalizable answers to big research questions relevant to the Foundation's strategic priorities. The grant is covering the salary for 3 full-time positions and an additional third-time senior staff member to support a series of research projects completed over the course of 3 years. Grant funds will also support data collection costs and honorariums for researchers serving as consultants and helping with project activities. We have 2 primary goals for this project. First is to investigate JTF-inspired research questions in global diverse samples. This will involve 4 individual empirical research projects carried out with the PSA network. The second goal is to investigate researchers' ability to predict generalizability, specifically that of the focal effects being studied in the 4 projects. The 4 individual projects are being selected in response to special calls for proposals. The first call was released in April of 2022 and closed in August. The second call will be released soon. To be eligible for consideration, projects must investigate questions related to 1 or more of the strategic priorities of JTF. These include the dynamics of religious change, intellectual humility, religious cognition, the science of character virtue, and health, religion, and spirituality. For each study, we will collect participants from standard university samples and from more representative community samples. This will allow us to more comprehensively examine the generalizability of the selected study's findings. A meta-science project will examine researcher intuitions regarding the generalizability of psychological effects. For each of the 4 selected projects, we will survey researchers in the PSA who are project contributors and those who are not prior to the start of data collection. Researcher beliefs and predictions about effects will be examined in relation to the research results to determine whether researchers can accurately predict the generalizability of the studied psychological phenomenon and what characteristics of researchers increase or decrease the accuracy of their prediction. These projects offer many opportunities for contributions from researchers across disciplines, regions, and career stages. First, I invite you to submit a project proposal to the second call for studies that will be announced very soon. Unlike previous calls, this call includes the option of submitting a short initial proposal due in February that will be considered for study incubation. To increase the competitiveness of proposals from researchers who are underrepresented in psychological science, authors of submissions selected for incubation will be provided additional guidance and support in preparing their full proposals. Full proposals, which resemble stage 1 registered reports, will be due in May. Regardless of whether they were part of study incubation, all full proposals will be given equal consideration during the project selection process. This process includes several rounds of review overseen by the PSA Study Selection Committee. Another opportunity for researchers is to join the 4 selected projects as collaborators. Data collection teams will be asked to make reasonable efforts to collect data from a certain number of participants and complete other project tasks such as manuscript review and approval by requested deadlines. Projects may also seek out additional collaborators to fill certain project administration roles. The most needed support will be in translating study materials from English to the sample languages. Thank you to the John Templeton Foundation for funding this research. And thank you to the Association for Interdisciplinary Meta-Research and Open Science for this opportunity to present. I welcome any questions live in the session or by email. Thanks for listening.
Okay, thank you very much. So great set of talks. Are there any questions from the audience? Yep.
I'm sorry, I can't remember the gentleman's name. He was with Unfold Research.
Yep, Dragan. Did you want to come up just to speaking to the microphone too, just so everyone can hear?
What was his name?
Dragan?
Hi, my question's for Dragan from Unfold Research. I'm curious if he's had any interaction with the Hypotheses platform, which is just a generalized annotation platform for the web, and if there's it might be helpful for me if he could talk about the similarities and differences between Unfold and Hypothesis.
Hello, just a test. Can you hear me?
Sorry. Can't hear you. So.
Is this computer looks slow? Yeah.
Try to unmute yourself again, Dragan.
Hello, can you hear me?
Oops. It's not coming.
I can unmute when you talk. I don't know what's going on with this. It's beautiful here.
Give us a second. Hold on just a minute.
You can like try this one.
Just getting the AV team sorted, and then
I know what to do. Let's try that.
I want to know. Now we're just getting people.
Oh,
So,
okay.
Can you try again, Dragan?
Yes, can you hear me?
Yes, that works. Thank you. Do you remember the question?
Yes, and thank you so much for your help, and thanks for the question. Yes, we're familiar with hypothesis. It's been present for a long time, and the kind of main few differences are that we'll be trying to collect very semantically narrow content. So while hypothesis offers the ability to post very unstructured comments without much of the voting interactions or anything like that, our focus is on collecting precisely and knowing exactly what kind of content are you posting, so that we can also parse that content and make the bidirectional connections between the content and use that to then create the graph. So the structure of the content will be more narrow, and that will enable us just to extract more metadata about the connections. Thank you.
Got a thumbs up from the audience on that. So did you have a question at all?
I did.
Yep. Got another one.
I had a question for Jill. I was struck that only 86% of the projects in what I think is a flagship effort have the materials available. So could you just say a bit about that? Why are the materials for the 14% not available?
So if I heard correctly, the question was, why was the material not all available?
Correct. For, I think you said 84 or 86%.
Yeah. Some of it we couldn't actually open. We had access to Inquisit and a variety of things, and so some just wasn't, we couldn't access it, wouldn't open anymore with it, and that was probably the big one that we couldn't do. It's just we couldn't actually open it at all in any sort of thing to look at it. I don't remember now offhand if anything was truly like there were no files there, but it was more a situation of being able to access and open what was there. Or sometimes, yeah, I think that was the big thing.
Yep.
Except for the consent forms. The consent forms were just missing.
Yeah.
But in terms of the materials, there was at least something there, I think, for every study, but whether or not it was accessible. And like I said, sometimes it was just literally the stimuli, but nothing around it.
And just on that note about the consent forms, so just for clarification, you mean the consent forms for the replications themselves, not for the original studies?
Right. Yeah. But they were all like in the reproducibility project, they were just doing the original, like they were just redoing a study, like a single study. Like this wasn't like a many labs where there's like 30, 30 minutes, 10 studies. These were just individual studies. And yeah, so we did contact OSF when we were writing the grant, and they said, yep, they would make sure those were there. And they sent out a request to the original people to ask them to please the replicators to put up their consent forms. And when we got the grant, and then we went to evaluate it, there were only 32 consent forms.
Fascinating. Well, great effort regardless, and interesting future work to explore from this project. I have a question for Kathleen. So just, I might have missed it. The process involved of selecting the studies that you'll be running for this first wave of the project. I noticed the themes of I think 3 of the 5 studies were based on some religious element. And I was just wondering how that came about. Was this, just a bit more information about why these ones are being selected.
Yeah, so that is based on the grant we received. So we're selecting studies that are on any of those themes. And so we are rounding down our first call of studies, and results of that might be announced hopefully soon. But we will be likely selecting 2 or 3 more on top of that. from the second call of studies. And generally speaking, these can be interpreted very broadly, but are related to the strategic priorities of the funding organization.
Yeah, I see. Well, good luck with all the studies. Sounds like a fun, exciting endeavor.
Yes, lots of work, but should produce some cool results.
Yeah. And I thought just one question to Kayana, who I see is also online. Just curious to know if you have any hypothesis as to why you noticed the downward trend in the randomization, number of studies with randomization and blinding.
Yeah, that's a very good question. This is still a preliminary result, so we're doing a lot more analyses specifically to look at specific areas, if there was certain types of studies that had less randomization, studies that use certain types of animals. So that's definitely an area that we're interested in as well, since that's definitely not the trend we'd like to see over time. But yeah, still something we're looking at. Not sure why, but hopefully will in the future.
Yeah, definitely interesting. Any other questions from the floor? No? Well, thank you everyone for joining, and thank you so much for joining live. We did not expect that to happen given the time that you're all presenting from. So we really appreciate you having being here live to take our questions. And so hope to see you at other sessions of the conference online. So thanks very much.